Thank you all for being here after the Bouillabaisse feast last night. And let me say Dzień dobry in honor of the Polish contingent that we have. And we all thank the organizer, but I have a particular debt of gratitude to Piotr, who did really a lot to make us possible for us to come here with our children. So thank you very much. So I will not talk about work that I did with all of these people, but they all helped me thinking through some of these problems, so I think it's important for me to acknowledge them. And in particular, I want to acknowledge Gozia, who has, in my view, had also a very important role in organizing this meeting. So many of us have visited with her, or she has visited us, and she has been very stimulating for us. She was the only one who managed for me and my husband to have a paper together after 20 years of working in statistics. So we've heard about the fact that p-values are somewhat under attack by Yoav yesterday. Here is another way in which they are perhaps a little bit under attack in the present world. We have a lot of data, and the way we collect data nowadays is comprehensive, exhaustive. So we collect much more data than what our original questions might be, and there is definitely this movement of that it's maybe a good idea to first collect data and then let it speak, ask questions later. And I think it's difficult to argue with the fact that more data is better and that we should look at the data to guide our intuition, but clearly we have to change a little bit the paradigm with which we do statistics and inference. And I want to talk about this from a viewpoint of genetics, which is the area where I work. In particular, I'm interested in gene mapping, which is, generally speaking, the idea of finding which portions of the genome are important for traits of interest. And now this question can be phrased at different degrees of specificities, so you might want to find which genes are important, and understanding which genes it's important because it tells you which protein, so it gives you an idea of the biological pathway that it's involved, and these are important for drug targets. But you may also want to know exactly which specific genetic variation in the gene is important, because that's what you need to know in order to do effective genetic counseling. What happens more and more nowadays is that you don't look just at one trait, but you look at a large collection of traits, and you want to see what genetic portions these different traits share. And so you are looking at this variety of questions. Now, gene mapping was never hypothesis-driven research. By definition, you go and look for something that is important for a trait, and you don't know where to look. There has been few exceptions with the candidate gene studies that have been actually not very successful at all. But even before we were able to explore the entire genome, in statistical genetics, people were very careful about multiple comparisons, even implicit multiple comparisons. So even when you were able to just go and look at a couple of proteins or the like the rhesus factor or the blood groups, people said that in order to see a significant association, you were to look for a very small p-value, because the a priori chance that whatever mechanism you were looking for was in those few loci in the genome, it's very small. And as time goes by, we have genome-wide scans became available, especially linkage scans, and there, in this context, we were looking at monogenic diseases, and the idea of controlling family-wise error rate became the general paradigm. For example, some of you may be familiar with a paper by Lander and Klurliag that codified different significant levels for these LOD scores. The landscape has changed again now that we are able to look not at microsatellites or 1,000 markers or 10,000 markers across the genome, but up to 1 million of them. We are looking not for monogenic diseases, but we are looking to understand the genetic basis of complex traits. And complex traits or polygenic traits mean traits that are influenced by very many different loci. So here I have an example of the polygenic traits par excellence, which has, in this study, they have identified 700 variants. So you know that the number of discoveries that you are looking at, it's fairly large. You're not looking for one place in the genome, but you're looking for many of them. The other characteristics of contemporary studies is that you don't look at one trait only, but you look at many traits. So for example, one study in which we are involved, that it's an example of all of these EQTL studies, your traits are the expression levels of genes. And depending on how you count and the sensitivity of your experiment, you might measure expression level for something between 20,000 or 30,000 genes. And in this particular study, you do this in 44 different tissues. So you have 20,000 by 44 different phenotypes, different traits that you are looking for. And at the level of genomic variation, you measure genotypes, in this case, 11 million SNPs. And your goal is to identify which variation in the genome influence the expression of some genes. But there's many ways in which you can rephrase the question. You can say, I want to find E genes, that is, genes that appear to be whose expression appear to be regulated by genetic variation. You want to understand if this genetic variation regulation is conserved across tissue, and you want to know which specific variants actually are behind this genetic regulation. Now, in these slides, which I'm sure the numbers are not very easy to visualize, to see for you, but it's a representation of all the tissues collected in this study. And I want just to point your attention to the fact that if we count in terms of E genes, so that is the genes that appear to be under genetic regulation, they identify 20,000 of them, 18,000, okay? So the number of discoveries that we have, it's large. We're not looking for one locus for one phenotype, but we have very many of them. Here is another study, an example from another study. This is exome resequencing, so we're getting all the variants in all the coding portions of the genome. It includes 20,000 subjects from Finland, and we have very detailed phenotype level for traits involved in metabolomics. So they use this technique, nuclear magnetic resonance, and in this picture you have the correlation between 64 of the traits that they decided to evaluate. Again, you expect multiple findings from all of these traits that you are looking at. So when you expect many discoveries, it is natural to want to control false discovery rate versus family-wise error rate. And indeed, for example, the field of EQTL has adopted the idea of controlling false discovery rate from its early days. What I want to point your attention and what my talk will be about is that there are many possible discoveries. So you can talk about discovering a genetic locus, you can talk about discovering a gene, you can talk about discovering a variant, you can talk about discovering all of these for one phenotype versus a group of phenotypes. And as Guillaume has so eloquently reminded us yesterday, FDR is a property that holds on average over the selected. So you have to pay attention about what your selections are. It's not that you can apply the same procedure and imagine that it controls FDR for all of these different types of discoveries. You have to sort of focus on what is the discovery that you want to report and make sure that the procedure that you are applying controls the false discovery rate for that particular type of discovery. Now this is has been noted a number of times, and I have here a quote from a paper by Sigmund and others that I think you have also referred to yesterday. And this is an example from an early paper in which I was involved, where we were pioneers and we control FDR for GWAS associations, making probably quite a bit of mistakes. So what you have here, it's on the x-axis, it's positions across the genome of different genetic variants, and on the y-axis, minus log 10 of their p-values. And this red horizontal line, it's a cutoff for significance that we established with applying Beniamini-Hochberg to all of these SNPs, to the p-value for all of these SNPs and number of traits that we had. So for example, on chromosome 2, you can see that there is a bunch of SNPs that pass this threshold, so we would reject all of these null hypotheses and say that we have an association here. Now, geneticists would interpret this not as having us found 10 different association results, but just one locus. And if you interpret, and what happens is the Benjamini-Hochberg procedure controls the FDR for the single hypothesis that you test, but not for this locus discovered. So if I look at how many loci I discover here, I discover this locus and also this locus over here, and maybe this is bogus, and this SNP could find its way in because at this particular locus that might be true, I had 20 discoveries, and so I have as is so nicely said here, inflated my denominator of total number of discoveries with a bunch of them that might be true, but they are really representing the same scientific thing. And this was also noted in the context of MRI studies and how you interpret the results from MRI, and Yoav and Ruth Eller have a nice paper early on this topic. Let me present you the same result from a slightly different viewpoint, that is one that we are particularly concerned in our context. Imagine that we are testing the association between a number of variants and a number of traits. And imagine that this is the true state of the world, where white means that we have an association and red that there is not. And so I have about, I don't remember, about 100 tests represented here with the small little squares, and say that I do calculate some p-values with a simulation and apply BH to all of these tests. And these are the rejections that I obtain, where a white square means that I have a rejection and the red squares means that I don't. If you count the false discovery proportion in this particular realization, it's below what is the average expected value, that here I think I set to 20%. But now if you look instead at the discovery of variants, so if you say, I want to see which variants are involved in any of the phenotypes. So you say, I'm going to look column by column and see if in this column I have at least one rejection, which would mean that this SNP does something, okay? Now, if you look at this from this viewpoint, you'll see that you have a false discovery proportion that it's much higher, because these errors are spread uniformly across the red area, and so I can discover a lot of SNPs, and if I count at the level of SNPs, this increases my false discovery proportion. And the same phenomenon has been noted in this p-filter paper, to which I will come back a number of times. Okay, so what is, I think, very exciting about working in genetics is actually the people involved in gene mapping are quite sophisticated statistically, and they have noted these problems. So if you look at the EQTL literature, they do control false discovery rate, but they don't supply all the p-values from all the tests to the Benjamini-Hochberg procedure or the story procedure that they often use, but rather they say, okay, let's first discover which genes appear to be controlled. So you aggregate the results from all the p-values for one particular gene, and you do your Benjamini-Hochberg procedure at the gene level, so that you avoid this problem of reporting results at the gene level versus conducting the test or not. And in this we have a paper submitted on this GTEC data, and I had nothing to do with this, what the analysts did when they wanted to say, now I don't want to simply know which genes have some genetic regulation, but I want to know which particular variants are involved in that genetic regulation, they used the BIC criteria, but their stopping rule was effectively implementing a selection adjustment. So they'll say, for each gene that I have discovered is regulated by some genetic variation, I'm going to do BIC to see which particular variants are important, but I'm going to stop adding variants to the model as soon as the p-value, the marginal p-value for this variant, it's larger than the p-value I required for genome-wide significance. So they are effectively doing a penalization to account for the selection.
I missed what is
sure.
EQTL.
EQTL, it's the quantitative trait locus for an expression, for a gene expression. Okay, so genetics is, I think, a playground for selective inference. There's multiple talks about selective inference here, so I'm not even attempting to do a review of that. Let me just talk about two examples of the work that we have done. One that relates to testing hypotheses on a tree, another one that instead relates to knockoff and but in both cases, what we are trying to do is to control FDR in the presence of structure and in contexts where we want to report discoveries at different levels. So let me start with this idea of testing hypotheses on a tree, and we are going to see a number of these pictures. So we have this finer scale, and then we have higher levels, I'll call them here, and each hypothesis at one level is parent to a family of hypotheses as the next level. And in my graph, a circle is used to indicate a true null hypothesis and a square to indicate a false null. And we're going to say that a parent hypothesis is true only if the intersection of the children hypotheses are true. So what does this have to do with what I've talked about? Well, for example, in the case of GTech, you can think of a tree of this type, where at the very bottom, you have testing the influence of a given SNP on a given gene in a given tissue. And then at the higher level, you might ask only if this SNP is important for a given gene, irrespectively from tissues. And at the higher level still, you might ask if this SNP is important for the expression of any genes. And I'll say that these ones are in some sense the most interesting discoveries that come from EQTL studies, because these studies are done to try to annotate the genome and to give a meaning to variations that we don't know how to interpret. So if you can identify that one particular variant is important for the expression of some genes, that's sort of the most important finding. Okay, and say that we want to do tests in this context, what are we going to assume? We are going to assume that we have p-values for the hypothesis at the finest scale. And then we can also have we can have any other p-value of our choice for this hypothesis at the higher level. In particular, we can also say we can obtain the p-values for this intersection hypothesis combining the p-values from the children. So here I have written out the Symes rule, but you can use whatever rule of your choice. Okay, so we are looking at hierarchical testing, and this is an idea that Dani Yekuteli worked out in a very nice paper in 2008. He probably had the idea before and applied actually to some genetic context earlier, but let me give that reference. So what you do is that you start from the top level. This H star here, it's a fictitious hypothesis that I put there just to make it look like a tree, so that it has one root node. But this is the first level of hypotheses. You test them, and say you might reject a couple of them that I indicated with a red boundary. Then you go ahead and test only the family indexed by this hypothesis that you have rejected. So at this second level, you are not going to test this particular hypothesis because it's not rejected by, it's not indicated by a rejected hypothesis at the previous level, and then you'll do your rejections, you'll move on to the next level, testing again only the family indicated by the rejected hypothesis, and you'll have some rejections. So this is what I mean with testing along a tree of families. Now, how do you count discoveries? You can say all of the rejections that I have are discoveries, or you can say really I'm interested in discoveries restricted to each level. So I'm interested only at the level 3 discoveries, or level 2, or level 1 discovery. Or you can be interested in what are called outer node discoveries, that is the discoveries at the finest level of resolutions that you can achieve. So here, for example, we were not able to reject any of the children hypotheses, so this one will be our outer node discovery. And again, Dani laid out all of these different ways of counting discoveries and described strategies in which you could control the FDR associated to these different counts. And his work dealt with 3 FDR, which is counting all the discoveries or level specific or outer node discoveries, but the strategy that he presented worked only if the p-values were independent across levels. Now, Beniamini and Bogomolov had a similar consideration, but if you will, they restricted themselves only to 3 that had only 2 levels, and defined a new notion of average error rate over the selected families. And what we're going to do is extend a little bit this framework, so I'll talk about it a bit more. And I think Ruth Eller has also another paper in the similar framework, but she'll talk about it presumably later on this morning. Some of the similar ideas are in the p-filter approach by Barber and Ramadas, even though that one is not for hierarchical testing, but it focuses on level-specific FDR control, and it's restricted to some specific ways of combining p-values. Anyway, so what is this new notion of what I'm calling selective FDR, which might not be the best name, but let's stick with it. So if you start at level 1, and at level 1 you consider all the rejections that you have, and you calculate the false discovery proportion, and what you tend to control is the expected value of this false discovery proportion, so this is simply FDR. When you move to the second level, you work family by family. So within each family you calculate the false discovery proportion, and what you aim to control is the average false discovery proportion across the families that have been tested. So I don't care about what happens here, I didn't test this one, but I'm calculating the false discovery proportion here, the false discovery proportion here, average them, and I want to control the expected value of this average. So when I go to the third level, I have the same situation. Now I have 3 families that I have tested, 3 false discovery proportion, I do their average. Formally, you can write it this way, so I have the average of the false discovery proportion, and then I want to calculate their expected value. As I said, at the first level is just equal to FDR. At the second level, you have the criteria that was defined by Beniamini and Bogomolov, and whose work this is an extension of. And generically, the average of the false discovery proportion that we are considering is this one, where if you look at it attentively, you'll see that the weights in this weighted average depends on how many rejections you had along the path that get to that family. But that's maybe. I am using the shortcut notation here, so I don't have max between 1 and the number of selection. And with S, I indicate the number of the selection set at each level. Okay, so it might seem like a strange error rate, and it seems strange to me at the beginning, but I actually think it's very handy and very nice. It allows you to make statements on discoveries at each level, because you have one of these ones for each level. It does incorporate automatically the order of testing, so you don't have to impose other coherence guarantees. There's no multiplicity across level that it's unaccounted for. One thing that it's true, and then when you do testing this way, it might happen something like this, where you have a discovery at a coarser level, it doesn't have to be followed up by a discovery at finer levels. However, if you have a testing strategy that guarantees that to be the case, then you have another extra consistency, so that if you control this selective false discovery rate at a lower level, okay, say that I know that I control here and I have consistency across level, then I know that I control also all the higher order false discovery rates. This is the algorithm, and it looks horrible. Let me just point your attention to these 2 lines. What we are going to do is that family by family we are applying BH procedure, but if our target level, say it's for each, our target it's Q, which can be different for each levels, what we do is that we make that target more stringent. And we make that target more stringent level by level, adjusting for the number of hypotheses that we have selected at the previous level over the number of possible hypotheses. Okay, and so here I'm saying it more in words, forgetting the fact that you have multiple levels and you have to combine them. The idea is that if you start with m total hypotheses at one level and you select only s of them, at the next level, if you want to control FDR at the level q, you'll do Beniamini-Hochberg, but with a level q multiplied by this penalization. Okay, and this is Marina's Bogomolov theorem. So suppose we have a procedure that control FDR within each family, and as what we are going to assume is that across path, the p-values are independent. So we don't need independence across levels, but we need the p-values for this branch to be independent from the p-value of this other branch. And it's not terribly clear that we need this, but the proof that we have at the moment has this assumption. Okay, so these are the two assumptions, and then if you follow this strategy, you do control FDR. Now, you don't need to control FDR at each level. Instead of having sort of FDP, the average FDP, you can have the average of any other error rate that you might be interested in. It's represented as an expected value, and you can do a similar corrections to get the same level of control. Okay, let me give you one example that I call didactic in the sense that it's not realistic, it's just given to show the idea where is this procedure different than others. And in this case, I'm going to represent my hypothesis in a matrix, and let's say the first level discovery, it's equivalent to discover that there is something happening in one row. Second level discovery, it's discovering that there is something happening within one row among the columns that are separated by these vertical lines. And the third level discovery is the discovery of the single hypothesis. So what you can see in this setup, where I colored in red the non-null hypothesis, is that you have a class of families that are much larger. So this family number 6, they have 90 hypotheses, versus these ones, they only have 2. You can also see that this one family contains many non-nulls, so that if you have a powerful enough procedure, when you're testing the single hypothesis, you have 90 good hypotheses to reject here. And so if you make a mistake and reject this one hypothesis, if you calculate the FDP across the entire area, that FDP might be fairly small because you have 90 good rejections to make. But if you look at the family-specific FDP, in this family, for example, if you reject wrongly this hypothesis, your false discovery proportion is already 1/2. And then you see also that you have families that are homogeneous, so that you might have an advantage in testing within families. And here it's a few methods compares, and we don't have time to go through all of them, but BH is applying the Beniamini-Hochberg procedure to the collections of p-values for all of these hypotheses for getting any structure. The method I described for you is this 3BH. And what you can see is that if you look, now I have levels, level 1, level 2, level 3. BH, it's supposed to control the level 3 FDR, and in fact it does, but it doesn't control these higher level FDRs. And what our procedure is supposed to control, it's supposed to control the FDR for level 1, and it does, and it's supposed to control the selective FDRs for level 2 and 3, and again it does. Now if you look at power, the power is comparable to BH. In fact, for level 2 and level 3, we have higher power, and that's because we are testing families of hypotheses that are homogeneous, so we can learn the specific characteristic of that hypothesis. Okay, we have serious simulations in the paper if you are interested. Let me just give you a sense of how does it result when we apply to this GTEC data. This data set has a small number of subjects on 44 tissue, not all the subject in every tissue. You already know that we measure about 20,000 genes, and the way we organize the testing is that at level 1 we want to discover SNPs that have influence on the expression level of any gene. Level 2 we want to specify which gene, and level 3 we want to specify which gene in which tissue. What we compare here is this 3-level procedure with applying BH to, say, the entire set of p-values, or BH applied tissue by tissue. Okay, so you have 44 tissue. You actually can do an analysis tissue by tissue, and here you have numbers, but let me point to some of the characteristics. So here I have BH separate, which is, you might argue, doesn't really control for all the multiplicity, but it's in fact fine because you have discoveries in every tissue, so you have a decent overall performance with our 3-level procedure. And let's focus on the discovery on the brain tissues. What you have here is the number of tissues in which one SNP gene pair was considered significant. What you can see is the 3-level procedure has on average a higher number of SNP gene pairs that are important across tissues, while analyzing tissue by tissue tends to increase the number of SNP gene pairs that you find that are tissue specific. And here you have an idea. Let's focus on this one that we see well. These are arterial tissue. You have, if you do the analysis separately, 6,000 gene pairs that seems to be SNP gene pairs that are specific to arterial tibial tissue, but that number decreases to about 2,000 if you instead use our procedure. So we tend to find more SNP gene pairs that have that are important, more SNP that are important for regulation of the same gene across tissue rather than in a tissue-specific fashion. Okay, so we got used to these refreshing moments. This is where I was last week over Cortina, now we can move on to the other approach that is looking at a similar problem but from a different perspective. For convenience, let me assume now that I have a phenotype and some SNPs that I'm looking at, and let me assume that there is a linear model and the error are well behaved, and all what I care is that beta is sparse, and I'm also going to assume that n is larger than p. And when I say for convenience, I really mean it. We are going to use knockoffs for doing this analysis, so we really don't need this assumption, but the way we have so that we don't spend time discussing what is a true discovery and what not, and to go fast, let's assume that this is the setup. What we want to do is to understand what is the support of beta, so we want to find the elements of this vector beta that are really different from 0. But again, what we're going to say is that if we imagine that the columns of X are different SNPs, we're going again to interpret our discoveries at different levels of resolution. And the way we represent this different resolution now is using this partition. So we have different layers, and at each layer I'm going to group the P variants that I have in a set of GL of groups. And what we're going to think is that if I make a selection among the betas, which are different from 0, this selection of which element of beta are different from 0 induces a selection at this layer too. Okay, first of all, let me try to explain why I might want to do this. So this is a study in which we were involved. It's a resequencing study. They targeted 13 or 17 loci in the genome that seem to be important for some traits. And so you have here the name of the locus. Then within locus you have a number of targeted genes, and within these genes you have a number of targeted variants. And the idea is to really try to understand which are the important variants, but also which are the important gene and which are the important loci. So if we go back to our tree setup, we can think that at the bottom level we have the genetic variants that are influence the phenotype, but you can group variants by gene and you can group gene by loci. And the framework for testing that we have in mind now, it's not a hierarchical testing, but it's a framework where you really make selection at the finest scale. Okay, these are represent the different variables that I put in my regression. But I am aware that if I make selection at the finest scale, this induces selections at this coarser scale. Meaning that if one of the hypotheses here is rejected, all the parents of this hypothesis are also rejected. So if I find that this SNP is important, it means that this gene is important, and it means that this locus is important. So this is the different way of going through this tree that I'm going to consider. And I didn't say it, but I'm using the tree picture because that's what we used before, and I think it's easier to compare backwards. But in this case, I don't need hypotheses to be organized hierarchically. These different layers don't need to be nested as long as you have partition in all of them. So what we're going to do, it's again, we discussed that there's many different criteria of FDR that you might want to control. In this case, we're going to focus on level-specific FDR, but we are going to use this, think about strategies like the one that I have described, where the rejections are, each layer are consistent by definition, right? Because you start making rejections at the finer scale, then these induce at the higher level. And we want a procedure that would control the FDR for the discovery at all of these levels. So I want to make sure that when I report these SNPs, I have an FDR control at the level of SNPs, but I also have an FDR control at the level of the genes that I discover associated to them, and an FDR control at the level of the loci. Now, to do this, we capitalize on a series of strategies proposed recently, and you'll see that there is a lot of Rina Foigel-Barber in these strategies. While putting them together in describing a procedure, it's a fairly obvious step. I do want to underscore that actually proving that putting them together works for what we want to do. It's not trivial. You cannot really adapt the proofs in these different papers, and this is work by Eugene Katsevich, whom I'm very fortunate to have as a student. So let me give you a very high view of what the strategy is. This time we build on the knockoffs, and Emmanuel opened with the review of the knockoffs. What we have to do is that we have all of these different layers. We are going to construct knockoffs for all of the different layers. What it means is that at each layer we are going to construct knockoffs that have the knockoffs for the finer scale are the normal knockoff, and the knockoffs at the higher orders have a little bit less stringent exchangeability conditions so that you can have a little bit more power. And then we use a system to explore what might be relevant variables at each layer, and you can think about the group lasso if you want as a strategy that uses a penalization that is coherent with the groups, and we get knockoff type statistic for each of the layers. Now the rejection set, what is interesting in is that, so this index L corresponds to the layer, and these knockoff statistics are for each layer, the knockoff statistic for the group. What we want is an index set where the knockoff statistics belong to the rejection region for every single layer. And we try to find a rejection boundary T that is this time a L-dimensional object because you have L layer, such that the estimate of the false discovery proportion, the estimate of FDR, it's controlled in every layer. So this might be a picture for a two-layer situation where we have a knockoff statistic for the single variable and a knockoff statistic for the group variables. If you were to do simple knockoffs, say you would reject when this knockoff statistic is large. So you have this dashed light blue line. This would be the rejection region for a single layer knockoff. What we are looking at is that we want to make sure that we have control of the false discovery, not of the single layer only, but also of the group layer. So we have two thresholds. We have these solid blue lines that represent our threshold, and this one is the threshold for the single variable layers, and this is the threshold for the group layer, so that the rejection region is only this upper left corner. Okay, so you have situations like the p-filter where you have many of these thresholds that you're trying to control, and you're trying to find a particular value t such that the estimate of the FDP is below that t across all the layers. Okay, and so this is the Eugene theorem, and I'll have to say that if you look at it, you see that we actually don't prove that we control exactly the false discovery rate. We control the false discovery rate up to a constant level, and of course you could rescale the procedure by this constant level, but in fact this appears to be more an artifact of our inability to prove a sharper result than something real. When we do simulation, it seems that this factor C is not relevant in practice. It comes, this factor C, from the fact that these knockoff statistics can be dependent from each other in any possible ways across layers. So you have quite a high degree of freedom, and using the same idea, Eugene was able to generalize the p-filter that has a similar flavor, but where you have to combine p-value using the Symes procedure. It's really fairly key to the way in which you run the test statistics. Okay, so because of this C thing that let me just show you a simulation where we run the procedure without ignoring this C constant. Let me just show you that if the target level is 20%, the multi-layer knockoff filter, which is the solid blue line, does control the target level. Okay, so this is one example of the fact that we don't quite meet that C. And what are we comparing here? We're comparing multi-layer knockoff with the original knockoff defined only on the individual variables, you have two sets of simulation, one where we vary the signal to noise ratio, and the other one where we vary the correlation between variables. What you can see is that the ordinary knockoff fails to control sometimes this group FDR, while the multi-layer knockoff doesn't, because it's designed for that. And this comes with a pricing power, but not a huge one. In particular, in this case, you see quite a difference in FDR and no difference in power. Okay, and let me conclude with one example from genetics, which is specifically the study that I mentioned before. In this particular setup, we have about 5,000 individuals and 700 genetic variants grouped in 85 genes. So what we are going to do is to use these two layers, one layer for the single variants, one layer for genes, and we are going to target an FDR level of 10% at both layers. And we are comparing our results with those that we would get with knockoff if we just target the SNP knockoff level. And you can analyze this data in many different ways, and we have done it our own, but at the moment we just want to focus on what's the difference of adding this extra layer of control. And so this is the same plot where we are looking at the knockoff statistics, and the solid blue lines are the boundaries for the multi-layer knockoff, and the dashed blue line, light blue line, is the boundary for the univariate, for the knockoff that focus only on the SNPs. And we have color-coded these dots that corresponds to discoveries on the basis of literature information. So green it's good. It seems to be a good discovery, a true finding. And, you know, luckily, most of the greens are in this upper quarter. If we compare the knockoff based only on individual variables with the knockoff based on the groups, you can see that there is this shift of the boundaries. And shifting the boundary this way, what does it mean? It means that we're going to lose these red dots, which luckily are presumably false discoveries. So in fact, putting a higher threshold and saying we do want to control the false discovery rate of the genes helped us to note that these particular variants that seem to be associated are probably not that associated. And that's because we are looking at the overall level of their gene. And to be fair, we do lose one variant that seems to be probably actually a good discovery, this ApoE5. But, you know, we were kind of very unlucky because it's right on the boundary, so it's not that we have a huge loss in power. Okay, thank you very much.
Any questions? Yes.
Briefly, how many layers you usually need or use?
In this case, we just use 2, and it doesn't, the price you pay doesn't really change too much across layers. So you really need to use as many layers as the level of discoveries that you are interested in. But we haven't used this in a large set of setup where to tell you, so that I can have really intelligent thing to do to tell you in practice. In our simulations, we mainly use 2 or 3.
In this discussion that's been going on in the literature, and I'm not following it that closely, so maybe you can correct me, on discovering rare variants, where there's an attempt to combine different markers within a gene in a way, say by multivariate analysis, to create an overall association of the gene, but where one doesn't care about so much, at least at the first level, about the individual markers. The question is the gene causative for a particular trait? Does your method say anything about that kind of problem?
So actually, this particular setup, we were motivated by exactly that problem, because we started with 17 regions that were already identified to be associated with some of these traits, and people did resequencing, actually hoping to find exactly the variants that were important. And the variants that might be important are rare variants, and when you do tests for rare variants, you have very little power. If you want to be careful, you might have, and you want to avoid false discoveries, you end up not doing them. So you end up doing this burden test where you aggregate the effect of many variants in the same gene. So you get a gene level response. Now, not particularly in this one, but in the hierarchical procedure I have described, you can use any burden test of your choice to get the aggregate, the p-values for the aggregate hypothesis. So it matches well with that. But my experience has been actually that these burden tests are highly unsatisfactory. They barely discover genes that you don't know that are already associated. And if you only discover the gene and not which variant, you really have a very limited increase of knowledge. So this procedure where you bring in the gene structure to some level, and you seems to me a way of addressing that, where you have a response at the gene level, but also you try to be more specific. I have to say that this procedure, the power of this procedure, it's limited by the power of the corresponding unilayer one. So we don't gain any power from knowing that multiple variants are in the same gene. We can only eliminate some false discovery, but in this particular procedure, we cannot gain. We can gain if we use the hierarchical setup in that I talked about before.
From your point of view, what mathematical methods, mathematical tools could improve such general approach to treating data? I have the impression inequalities in theory of functional inequalities.
Oh, that is a very difficult question for me to answer. Now, I try to hide, I'm not a mathematician in here, so what I can tell you and David and Emmanuel can respond a little bit more to what mathematics is involved here. The knockoffs uses martingale theory, and the proof for the knockoff controls is based on martingale. We could not use, or Jean could not use, the same proof for the multi-layer knockoff, because you now have, you would like to define a stop in time, but you have filtration. It's not obvious how to define a filtration for which filtration this stopping time is defined with respect to when you are looking at multiple layers. The P filter uses actually a lot of results similar to the one that Yoav and Dani Yekutieli had used for proving control of FDR in case of dependence. So it's sort of the same set of tools. But I don't know what other mathematics you might need to develop.
Just one last question.
For the tree-based approach, some of the independence assumptions you need to make, they seem to come from wanting to control the false discovery rate, whereas if you control the family-wise error rate, you wouldn't necessarily have to make those assumptions. Do you consider using family-wise error rate as well, or you feel that those are good concerns?
No, you're right, and we do consider using family-wise error rate. In particular, family-wise error rate becomes more meaningful when if you have the tree. Let me get to some tree. Suppose that at the end here you have some families where you don't have very many hypotheses. It doesn't make sense to target false discovery rate there too much, because what is the false discovery rate out of 4 hypotheses? It's kind of a meaningless thing. So it might be more useful to control family-wise error rate, and you can there are versions of the procedure where you can control family-wise error rate.
Okay, let's thanks Chiara again.
Thank you.